it can show that this make up better and also to invest in research. In particular, the genus Rubus of the family Rosaceae has been divided by Folk in 12 subgenus. That in each subgenus there are described 100 and more than 100 plants and varieties. So from the tropical to the circularctic, they are very spread. So everywhere in the world there are one or more wild species of Rubus. There was some studies that could see that there is some parental ancestral genotype that are the same. Raspberry is an heterozygosus, an heterozygosus diploid perennial species that have a basis chromosome of 7, and the habit of the raspberry plant is that grow in a shrub. That is renewed continuously by generate new shoots. The raspberry plant have a perennial roots, and these biennial shoots are divided into the first one that are called primocane that grow in taller in the summer, and the floricane cane or shoots that not grow very high in taller, but grow in short lateral branches, where it contains several leaves and terminal flower cluster that allow the formation of the fruit. There is two types of fruiting, so of variety. The floricane and the floricane that has the ability to create a new cane that is called primocane, like I said before, in the first year. So in the spring they grow, and in summer continues the growth. Under the autumn, it became lignified, so woody, and in the second year, when it's called floricane, it is able to generate clusters that contain from 6 to 8 flowers that are able to create enlarges the fruits. In the floricane production, there is one production each year for the first starting. After that, the floricane cane are died, but in the spring, during the spring of the second year, the plant are generating through the roots another shoots that are called primocane. So the start of the plant cycle is regenerate again. There is an exception, because there is also another varieties that are called 4-bearing or primocane bearing, that are able to produce a double crop per season, because in the autumn of the first year is able to generate the fruits. This is due of the chilling hour requirement that are very low. So after the fall season, there is the pruning of the tip part of the plant, of the cane. After that, in the second year, in spring, there is there is the buds of the upper part of the lower part of the cane that produce again leaves and flower and fruits. At the same time, as I said before, for the floricane, in the spring of the second year, they generate new shoots in order to obtain an increasing of production in the year one by one. The crop quality could be reduced in this case because the soil and the plant needs more energy to produce a double crop. But this is very unadvantageous for the prime cane fruiting, because we could obtain a double harvest due to the short chilling requirement. So it's and also the high temperature. So it's possible to grow this kind of plant in all the regions. So not only in the mountain, but also in the warmer region. And also, it's possible to take the pruning very in an easy way, because the upper part is the part that that I have produced it. So this is an aspect very important for our region and our climate. Okay. The raspberry produce a lot of flower. These flowers are self-fertilized, and the flower born in cluster on lateral stem. They produce they have a flower with 5 petals surrounded by a green sepal. This is not a real fruit, because it is a composite that is an aggregation of fruit made up of many droplets surrounded the receptacle. So when in every droplet there is one seed. The seed is the real fruit. The fruit of the raspberry matures in 30-36 days after the pollination. And when we pick the raspberry, we take the flower and the receptacle remain attached to the plant. So it's very easy to manage like this kind of fruit, because it presents an hollow core, so it's without any structure, rigid structure. Also, the raspberry plants have needed to sunlight exposure, and in order to ripe the fruit, and they prefer a good high circulation because there is a lot of the habitus of the plant is a shrub, so it's very poor the high circulation inside the plant. So in order to protect the plant for the attack of insect or attack of water, spring water, that could be dangerous for the plant. The soil needed is well drained and with a high organic matter, and the optimal pH is subacid pH around 5. And the high water is, the quantity of water is it's an important thing because for support the stress of the high temperature and the foliage, the leaf very vicine, insomma, very each other, very near each other. So it's an important implement to the high circulation and water needed for support the stress of temperature. The raspberry could be breed in a trellis system. There is the possibility to grow the cane in a wire that is around a bar. For instance, we could see a row system that is the whole system, the old system that take inside of a runner wire the plant, the cane. Another better system is the trelling of the tea stake, that is quite similar, but it's possible to be grown the cane of floricane in the in the more far wire, and inside it could be to grow the primocane that in the first period doesn't occur with fruit, doesn't produce any fruit. And the same of the tea stake is the view support, where, like I said before, the floricane are put in the part more easy to pick, because the good manage for the raspberry trellising is in particular for the harvest. The main aspect in red raspberry breeding program, of course, the market want to satisfy some characteristic of the red raspberry. So, of course, a good productivity. Repeat flowering can occur this, can satisfy this productivity, but also good vigor for manage and harvest very well the plant. Flower fertility, large fruit size, and high quality and resistance, of course, to biotic stresses. These are altogether the polygenic aspect, polygenic trait, because not only one genus is responsible for that, but there are a complex of genus that are responsible to the high productivity, for instance. The repeat flowering for our studies in the past that could I study is that the parental is the percentage of progeny obtained by the parental autumn fruity express this character only the 3-to-1 clonal material. So this is obtained because every genotype depends not only by that, but also it is influenced by the environment. In that case, the flower initiation, indeterminate buds, and cane elongation are controlled by the day length and the temperature in order to obtain a trait that is possible to see, so the phenotype. The good vigor is the most important plant characteristics because the cane is responsible to support the fruit production, and also is an advantage for the harvesting because it could be reduced the high cost associated with the hand harvest. Another important aspect could be the turn. The cane could be surrounded by turn or turnless or turn-free. This gene is the S gene that is responsible for the spineless phenotype. That could be an easy way when we harvest for the harvest. The production, the productivity of the plant, it's variable due to the variety. It's around 8 to 10 tons per hectare. And as I said before, the productivity depends for many other factors. So the flower fertility, the ability to set the fruit, the droplet size, the number of droplets per fruit, and the cluster per plant. Another important role in the market is the color. The color could be in the same species. There is a red, pink, yellow color, red raspberry. So we could be a different red from to intensity, a different intensity of red. The best color is the bright red. The production is measured by the weight of the single fruit. So a fruit is weight more than 8 grams is classified big, and the fruit with less grams than 2 are classified small. Of course, the shape is very important because they classify in a small, in a big fruit also for the shape, because a conical shape is more big, it appears more big than an oval shape, because of course we have to remember that the fruit are hollow cored, so the inside is empty. Another important aspect for the quality characteristic is the manage, so the easy to pick, because the raspberry it depends, easy to pick how to set the fruit, how to set the flower, so how many droplets are created and how they can adhere to the receptacle. Also the firmness, because when the harvest when the hand harvest is done, the soft fruit is very attached. Of course, it's possible to differentiate the destination of use of this fruit after this classification, because the fresh consumption is recommended, but the fresh consumption is only for fruit that remain entire, so in the whole part. Perfect. For many other that maybe have the effect of crumbly fruits, they are destined for the transformative form, like jam, yogurt, and ice cream, that of course are always important. We can measure the qualitative aspect of soluble solid content, so the sugar inside the fruit and also the acidity. But there is a lot of nutrient value that could be examined, and because the berry have a low calorie index, but they are rich in vitamin A, B, C that reinforce the immune system, and they control the cardiovascular disease. The good content of mineral salts like magnesium and phosphate that are able to control many reactions of our body. The good content of carotenoids, they are the precursor of vitamin A, and of vitamin A can offer an important fiber intake. The important fiber intake that could be an important role in our digestion, and also the high content of flavonoids, anthocyanins, so the bioactive compounds that could fight the action of free radicals in our body. At the end, there is also the resistance to insect and pathogens that are very present in this red raspberry species. One of the most disease is the raspberry spore blight that cause an evidence in the leaf of brown color that enter inside and pass it through the cane, and this is caused by a fungal that is called Didymella planata, and this in another study is possible to show that the spread of the Didymella planata is referred to the thorn. So if we select a gene with a thorn free, the diffusion, so the spread of this spore blight is less. Of course, there is also the gray mold that is a very high problem for the fruits. They became rich in mycelium, so they became brown and fall. The best solution for these things here is to remove the reduction of water, like after the fall, after the rain, and also there is the main attack by insect that could be another problem. The Antonomus rubi is the weevil of the raspberry that hit the foliage, the leaf, and also create our larvae in the soil and destroy the roots. Another one is could be the beetles of the raspberry that is the Biturus tomentosus, that the adult produce her eggs inside the flower, so the fruit is contaminated and the harvest is damaged. In conclusion, the aim of my study is create a new seedling that could be a positive result in the market, so satisfy the ever-bearing, so the ever-bearing parental genotype. Quindi I have to select a ever-bearing parental genotype, and with the trait of temperature, of the temperature and photoperiod, I want to induce the ripening in all of the seedling, and of course select a high flower fertility to obtain a good quality for the fruits and obtain a best harvest. And also I want to create the gene of turn less repeat in order to reduce any possibility of aphid and fungal spread. And of course, but not at least, at the end, not at least, the quality of the seedling have to be satisfied for the market. And so, thank you.
Bene.
Sorry for my English.
Okay, Valeria. Well, even if you ended your master in English, you need to
yes.
more relaxed in doing the presentation. Hopefully, this is a new topic for you, and so you did some work in trying to learning more the different terminology that you need for that. I don't know if some colleague has some question.
I have a couple.
Sì, prego.
Corti sta anche. Beppe,
Vabbè,
puoi andare tu?
No, prego.
Allora, I have a couple of questions, which is the first is this one. At the beginning, you show an equation that say that the phenotype is given by the effect of the genotype and the environment. Don't you think that something is missing in this equation?
Yes. I put only the main aspect, more in general, but of course there is I try to explain something about the polygenic aspect. So not the Mendel trait or transmission.
No, I'm not referring to. It's also Mendel traits are the same, have the same genetic inheritance of the others. So I think that this is a general equation that gives you the idea of what is how you can describe the phenotype. But something is missing in any type of equation, which is the interaction between the genotype and the environment, which is not the same of saying that the phenotype is genotype plus environment. Some genes have a major effect on the interactions, and they also are making an effect on the whole genotype. So this is the general equation is genotype plus environment plus genotype by environment interaction. So G plus E plus G by E equal the phenotype. The other question is about the breeding. Do you think that in raspberry it's relevant the use of genomics or other molecular and biotechnological tools or not?
I think that
really consider at all these aspects.
Yes, I think that could be very important for the resistance of biotic stresses, in particular for this aspect. But I didn't consider that because in the first top, I want to select something of good quality in harvest and in fruit. Yes, of course, if it doesn't remain safe for a long period, but it's for the high cost of market genes. I didn't explain something about it.
Okay, thank you.
I can say something about that. Also, we discussed a little bit this. I fully agree with your first question. About the second is something that, yeah, she may have introduced something in this seminar, but it was also the idea to use this part also in the second seminar related to the methodology. So going more in detail on this part. But okay, that was just a beginning, and also for us it's the beginning of the breeding program of raspberry, thanks to a collaboration with the company, and we agree with the company to focus particularly now at this starting point only on the standard traditional breed. So it's not a fault of Valeria, but it's not a decision.
No.
But that is a decision.
I think that
related.
I would say that it's. I agree with you. You don't need necessarily to. But I mean, nowadays, I think that if you speak about breeding, at least you can say, well, this is focusing on the, let's say, the phenotypic analysis or whatever. But I think that somehow a mention would be, even to say we don't speak about it.
For sure, it will be in the next seminar related to.
Because I have a curiosity at this point to know if there is, I mean, a major activity on that. There is anything done, or if the level is more or less the same of other species, or is still, let's say, in the early stage.
No, among berry, there are a couple of groups that are working specifically on raspberry, and they already developed different genomic tools for marker-assisted selection. Particularly, the James Sutton Institute in Scotland is doing that. Actually, they almost sequence the genome of raspberry. So, for sure, at genomic level, there are already also for raspberry many information available. Okay. That was my, sorry, I give the answer. Beppe, avevi una domanda anche tu?
No. I want to just say that probably English could be improved.
Yes, of course.
Yeah, that's just a sketch. I mean, are you at the first? Sorry, I don't remember. First or second year?
I'm the first year.
So you had time. Yeah, you had time.
Okay.
Good. Just this.
The beginning of the, and also I say that she has a master. She is from food technology, so I think that she is missing some of the.
Yes, I missed something last year.
Okay. Thank you.
Thank you.
Okay. If no other question, we can. Thank you, Valeria.
Thank you to you.
And then now there is Lorenzo Rocchetti, no?
Yes, here I am.
So you can, I don't know if you speak Italian or English, you can share your presentation and start.
Okay. Can you see the presentation and the audio is clear?
Yes.
Okay. Okay, perfect. So, good morning, everybody. Good afternoon, everybody. Today, I would like to talk about the current status and new opportunity for European food legume breeding. A particular focus will be put on chickpea genetic resources. So we live in a human-dominated planet, putting an unprecedented pressure on the Earth system. And the Sweden scientist Johan Rockström in 2009 described 9 planetary boundaries within human can develop and thrive for generations. In particular, according to Garden et al. 2020, agriculture is putting heavy pressure on 4 of these planetary boundaries, in particular on biosphere integrity, land system change, freshwater use, and nitrogen flow. And respecting these planetary boundaries, we can only feed 3.4 billion of people. So the question is how to feed the growing population. Well, we can operate in a safe zone, increasing the agricultural efficiency in terms of irrigation and cropland expansion, water, nutrient, and land management, reducing food loss, and in particular on diet change. And doing that, we will be able to feed 10 billion of people respecting the planetary boundaries. I would like to focus on the diet change because the International Panel on Climate Change wrote that the transition to novel plant-based diet is paramount in order to mitigate climate change. The same things was reported from Poor et al. In 2019 that they were saying that dietary change of consumers can deliver environmental benefit on a scale not achievable by producers. So this is a fundamental the consumer change. And I would like to come back to the planetary boundaries, and in particular, I would like to focus the attention on Europe. And according to the authors, Europe is particularly high risk for nitrogen flow because it's an area of intense agriculture, and so the nitrogen flow is a high risk. So what we can do in this scenario? In this scenario, food legume represent a valuable opportunity for feeding the growing population in a sustainable way. Why? Because they will be able to mitigate climate change because they will be able to reduce greenhouse gases, so they have low global warming potential because they represent a good source of protein that can be a valuable alternative to the meat and the dairy product. And also they will be able to reduce the nitrogen fertilization because they are able to fix nitrogen thanks to the symbiosis with bacteria. So among the main among the food grain legume, we simply remember chickpea, common bean, lentil, and lupin. And I would like to say that also the Polytechnic University of Marke is leading a project about the genetic resources for those crops. So what is the current situation for food legume in Europe? Well, Europe, respect the other developed country like Canada or Australia, reduce the area sown to grain legume, while those other countries increase. So actually, food grain legume are underrepresented in Europe. So only near to 2% of the total arable land is destined to food legume, while in the worldwide basis, it's around 14%. So the low investment in food grain legume in Europe reflect actually the lack of breeding effort for creating varieties that are adapted to the European agroenvironment. So, but something it's changing. So the consumers now are increasing vegetarian and flexitarian consumers in Europe. As we can see simply from the Google search trends, is increasing the trend of consumers that are interested in vegetarian and flexitarian diet. So they are interested on reducing meat and dairy products. And this is translated in a market, so in economic value. So we can see from the mid-report of Europe in 2018 that the meat alternative and dairy alternatives is gaining more market value, economic value. So what are the opportunity for breeding in Europe? So the large part of the calories product by for feeding the population now are mainly produced by 4 crops: maize, corn, maize, wheat, rice, and soybean. And as you can see from the paper of Kasman et al. In 2020, they show that the yield of those crops is linearly increasing, but the relative yield gain is decreasing. And in particular, in some area of the world, the breadbasket of the world, like Europe for wheat and China for rice, there is a plateau on yield growing. So there is a yield stagnation. And this offers new opportunity for the orphan crop like food legume, where breeding effort were not put it. And for example, we can see here the same things. The yield of cereal increased because of breeding effort and improvement of agronomic technique, while the yield of pulses remained the same. But we have an opportunity, and this is demonstrated by the breeding effort on soybean. So soybean was one of the green legume where a lot of breeding effort were put it, and we can see that yield increase at the same trend of the cereal. So there is an opportunity also for the other food legume crop. Chickpea is an example of among the food legume crops. It's a cold season green legume. It's a double diploid genome, self-pollinated crop with a draft genome around 800 megabase. And in particular, we distinguish 2 market type or cultivated type of chickpea. We have the Desi type with the dark seeds and purple flower that are mainly cultivated in the Indian subcontinent. Then we have the Kabuli type that they have light seeds and white flower. And so chickpea actually is an important green legume that is suited to the European environment and offer important agronomic characteristic like nitrogen fixation, as the other green legume crop, is a low input crop, adapting marginal lands. And as you can see also from the picture of our region, it's used in crop rotation with wheat in dry area. And also it grows with the soil residual moisture, so it doesn't need irrigation supply. So in order to create a European breeding program, we need to start from the genetic resources. And how to valorize that? We need a pre-breeding workflow. So everything starts from germplasm collection that has to be described at the genotypic level, phenotypic level, and gather as much as possible geographic information of the adjacents. Then we can develop population. So like a core collection or segregating population. And through the study of population development, we will be able to map in the genome trait of interest and select advanced genotypes that they can be used as a new varieties itself, or they can be used as a donor of a particular adaptation trait, for example, and they can pass the adaptation trait to the former elite varieties, entering a breeding cycle in order to release a new cultivar. So in order to analyze and collect the information from the germplasm collection, it's fundamental to study the domestication history. For chickpea, chickpea was domesticated in the Fertile Crescent as the other crop like wheat, barley, or food legume like lentil and pea. And in particular, the wild progenitors of chickpea are mainly concentrated in this area in southern eastern Turkey. And the gene pool, the germplasm is divided in 3 gene pool. According to Ladzinski, we have the first gene pool where we found Cicera retinum, that is the cultivated type, and the Cicera reticulatum, that is the wild progenitor that can be crossed with the cultivated type and give fertile hybrid. Then we have the second gene pool with Cicera equinum spermum that can be crossed with the cultivated type, but sometimes the hybrids are not fertile. And then in the third gene pool, we find all the annual species that actually offer meion constraint to the cross with the cultivated type. So among the crops that were domesticated in the Fertile Crescent, chickpea is an exception. Why? Because all the other crops and also the wild progenitor of chickpea have an autumn sowing, while actually now we know and we see that the cultivated type has a spring sowing. So according, because there was this shifting because the first farmer changed growing season, because in autumn sowing, chickpea was devastated by an important fungal disease that is called ascochyta blight. So the first farmer tried to change sowing season, and according to Abo, that was the major domestication trait, was the loss of vernalization for chickpea in order to shift growing season. So as we said before, we can collect wild material in southern East Turkey, like Cicer echinospermum and reticulatum. But nowadays, chickpeas is spread all over the world, and we have landraces spread in particular. So from southern East Turkey, the chickpea spread it also thanks to the movement of the humans, spread in the southern in the Indian subcontinent, where we can largely found the Desi type in Ethiopia, and also in the Mediterranean basin, where we found the Kabuli type. So here is where are located the land races, but nowadays chickpea is also cultivated in Australia and in America, because they start their own breeding program thanks to the land races, and they start mainly the breeding program on the Kabuli types. So coming back to the breeding, pre-breeding workflow, now I will talk about how to develop a population. So an example of population that is important to develop in order to study the germplasm is the core collection. So what is a core collection? The core collection is a collection of, is a reduced number of accession that represent the entire variability of the whole collection. So according to Frankel in 1984, he was saying that the core collection represent, with a minimum of repetitiveness, the genetic diversity of a crop species. And so I would like to mention that in chickpea, there is a developed core collection from ICRISAT that is a member of CGR. And then we can also create segregating population. So we can choose different genotypes, for example, from different geographic part of the world. We can cross them together and follow the recombination between traits, and in order to study the recombination and mapping in the genome of those traits. So just I would like to mention that in chickpea, I mentioned only the multiparents population. We have an AM population that was described by Shin et al. in 2019, and a magic population developed by ICRISAT, and an ongoing developing magic population from ICARDA. So we saw how to develop population. So now, how to map in the genome the trait of interest? What are the genomic resources that we have? So the genomic resources are fundamental for doing that and are part of the pre-breeding resources. And here I report what is available for chickpea. For chickpea, we have the sequenced genome of a Kabuli type, in particular of this accession. Then we have the reference genome for a Desi type from this accession, and the reference genome for a wild. But the technology is improving, and we know that there are some variants, some genes that are not present in all genotypes, in all individuals. So there are some variants that are unique. So it's fundamental to develop a pangenome, so that is it's a technique that allowed us to assembling different genome in order to study variant or gene that are unique and present only on particular organism. And the pangenome is ongoing development and for chickpea by ICRISAT. And so we can link all this information together. We can bridge the phenome to the genome. So we start from the genetic diversity, from core collection and segregating population. And then we have all our genomic resources from the reference genome, the sequencing of different genotypes, and the pangenome that is ongoing. And then we have the field trial. So we will be able to phenotype and describe the trait on field, but also all the omic data that range from metabolomics, transcriptomic, and proteomics. All this information together will let us implement gene discovering and breeding tool, from gene discovering like linkage mapping and genome-wide association mapping, and breeding tool like genomic selection that it will help us to select advanced genotype. So what are the current breeding traits that I mean, here I report some reference about the QTL that were mapped in the genome, were mapped for chickpea. So for breeding was important the seed weight, the drought tolerance, the Ascochyta blight resistance, plant growth habit, flowering time. But what we can foresee for the future. So there are innovative products like the pasta made entirely by legume flour. So there was published this patent from Mariani et al. So they are able to make an innovative product of pasta made entirely by legume flour, and this products open the door for new breeding traits in order to breed and to find genotypes for making quality products. So concluding, in the context of reducing agricultural input, and in particular feeding the growing population, food legume represent a valuable opportunity, in particular, especially for Europe, where nitrogen flow risk is high. So phenotypic and genomic characterization of the legume genetic resources is fundamental in order to start a breeding program for Europe. Chickpea is an example among the food grain legume that we can use, and develop a local breeding program for chickpea in this example will represent a valuable opportunity for Europe. Thank you for your attention.
Great, thank you very much. Great, thank you very much.
How to
There is it's a background. It's a background. Okay. Okay. I don't know how to stop the background. To the return. Wow, very interesting presentation, and yeah, the English is really much better. So congratulations on that.
Oh, thank you.
If there are some questions. So no, any question? Well, I don't see people. Well, from my side, I think that you gave a good introduction of all the idea of the program. And also I know that there is a big project behind all that. So you will have a lot of work. And about that. In that case, the molecular approach, it will be much more important. And I see that you already identify several markers that can be applied in the breeding programs. And well, as a general question, do you know that these markers are already in some way applied in different breeding programs? Is something new that you will apply in your program, in your activity?
So yeah, mainly the breeding for chickpea was focused, for example, in the CGR Institute, for example. They were developed many market trait. For example, it's famous the drought tolerance trait. So there is a market assisted selection for drought tolerant chickpea. But what I'm interested in my future process of the PhD is on the genomic selection. So the genomic selection we will be able to select genotypes also for polygenic trait like yield. So it will be interesting to develop these tools. There are already some reference about it, and it will be interesting to develop these tools here in our environment in order to be able to test some genomic selection methods here.
Well, genomic selection is a very interesting approach. So I think that for that it will be really useful and important, also for identifying the capacity of the different germoplasm in transferring these traits.
Exactly. Also for valorizing the germoplasm resources, it's fundamental the genomic selection in order to select some advanced genotype that can be used as donor for particular adaptation trait.
As a parents, so I think that it's really an important tool to better classify all the different parents, germoplasm identified parents to be used in your breeding program.
Yes, exactly.
That it will be the main input. Okay, thank you. I don't have other question. I forgot to say that I'm recording your seminars or almost since the beginning, also the beginning of Valeria's seminar. Sorry, I forgot to say and ask, is that okay? Do you agree in that? So it will be available for everybody. The same was also for Valeria, and the same it will be also for the next. Okay.
Thank you.
Okay, thank you. Okay, let's stop here. If no other question, if there are no other questions, next, I forgot the name. Lorenzo Camponi, is that true?
Yes.
Okay, Lorenzo, it's your time.
Yeah.
So it's okay that I keep recording also your seminar.
Yeah. It's not a problem.
Go ahead.
So, do you see?
Yes.
Okay. I'm the PhD student of the pedology group, supervised by Prof. Corti and Prof. Cocco, and the main theme of my research here in the university is the evaluation of soil suitability to management changes. So we'll start with a brief summary. If the presentation let me do this. I will explain you what the main concept of the land use change, the effect of the land use changes on the soil properties. Then we will see the importance of the pedology inside the subject of land use evaluation. And we will see very fast some kinds of surveys used in this subject, and then at the end I will explain you the research project, my proposal. So let's start. Land use change is a term that refers to two major processes. One is related to the expansion of the or contraction of the land cover. So, for example, we have a landscape patch that is pasture, and then during the years is transforming to forest. So this is a kind of land use change. The other kind of example is the change of management inside the same patch. So we have the same forest. We change the management from coppice to high forest. This is a land use change. Sorry about the presentation. Okay. The influence of the land use change on the soil properties is on the biodiversity, of course, of the soil, the physical properties like structure, texture, and others, and the chemical properties, so pH, carbon content, etc. In particular, the soil influences the management of the area, so the land use, and of course the land use could affect the soil properties. So now we see some example. In particular, for physical properties, I choose these two citations because are very clear example, and we can say that the influence on soil texture is a common characteristic of the land use changes. And in the second citation, I want to stress the concept of the contributes of the, for example, the macro porosity of the soil that is very few important in the volume of the soil. In fact, is less than 1%, but is very important for the hydraulic conductivity. In fact, we see here is more important for the water flow and influence more than the 50% of the water flow. Another example of the effect of land use changes on the physical property is the badland landscape that is widespread, and also in our country, in particular in our region, is a very common phenomena, and is due to the interaction of the land use on particular vulnerable substrates and soils. For the chemical properties, we can see that on an example of the changing from a natural ecosystem, a marsh, to cultivated land, in this case sugarcane plantation, and we see the influence on the principal chemical properties. For example, we see the soil organic carbon percentage, and we see a reduction in the changing of land use. We also see a reduction on the water extractable carbon and in the carbon nitrogen ratio, for example. And this slide is dedicated to the pedofauna, the biological component of the soil. So we have microorganisms, one example for microorganisms and macrofauna, and the other for other organisms of the soil that are the mites. And we see here the influence of these land use changes in different scenarios on the composition in terms of species and in terms of biomass, so in terms of quantity. In particular, we see in the second graph the differences between different land use management. So we have here forest, shrub land, old field, and two other kind of management, and we see the amount of different species in these samples, and how the different management could affect this kind of soil properties. Not everything is lost. I show some data that are not very good, but we have also to say that soils have a resilience, so a sort of inertia, so they are able to maintain their homeostasis. Also, if they are stressed, if they are subjected to land use change and change of management. And the take-home message that I want to say to you that is we have the necessity of better understand the effect of these changes on soil properties, because they are widespread and they are also very diffuse and very fast nowadays. According to this slide, I want to stress the concept of the importance of the pedological surveys in the land use change evaluation. So I will show you two tables just for said that the pedology is the core subject in land evaluation. For example, we have here the terms used in land evaluation and the correspondent term in soil quality evaluation. So you see that the measure of these terms are referred to the soil quality evaluation. And also in this other slide, you see the importance of soil parameters on the physical, chemical, and the visible attributes in the land evaluation. So you see, for example, physical attributes in land evaluation are referred to the measured soil physical parameters. So, for example, soil texture, bulk density, porosity, and so on. In our subject, we use a different kind of survey manual, and I put in this slide the four most important, the four most used around the world, and in particular the soil survey manual of USDA is like the Bible of the pedologists. You know, is a very complete manual for the survey experience. There are also to cite the FAO guidelines, because is a book that is more concentrate on the practical aspect, the productivity of the soils. So they are two main books that have a different approach. Here I put the cover of this book. There are also the last two that are one of the Australian agricultural department, and the other came from France. And now I want to spend a few words for the visual soil assessment. That is a different approach in soil surveying, is born in the FAO, so is a word spread approach, and is focused on the visual approach. So we can evaluate the soil with this these parameters, but with the visual approach. So we see the texture, the structure, the presence of the biological activity, the rooting dip, and so on, by a simple visual approach. And these are the soil scorecards that are very simple, and is enough that you put the values proposed from the scorecard, and you sum it, and at the end you will obtain a soil quality index that could be low or could be moderate or good. And you can have the assessment of your soil. In the right one scorecard, you can test also the performance of the species, the tree or the herbaceous species that you have inside your cultivation land. And the strengths of the VSA method are that is very quick and simple. So everyone could do in few minutes everywhere, and is a very good method, qualitative method for soil evaluation. Is focused on the producer, so the producer could do this kind of evaluation even if he's not a professor or a pedologist, a professional pedologist. So, you know, is a very good instrument. My project proposal is about a surveying, soil surveying, traditional soil surveying, and also remote sensing soil surveying in different productive areas. So we have chestnut forest, hazelnut plantation, and soft fruit plantation. And the focus of this project is the performance of this kind of species and the effect of the changing, the land use changing on the soil properties in different, in two regions, in our region, Marche region and the Abruzzo region. These are the three sites. So we have Pieve Bovigliana in Macerata province with the chestnut plantation, chestnut forest, and Fiastra with the hazelnut plantation. And in Abruzzo, in Teramo province, the soft fruit orchard. Here the principal necessity of this species, and we have the hazelnut with parent material, kind of parent material from marls or sandstones, and the texture that is from silty clay to clay loam. So the hazelnut needs also very good water contact in the soil, and the pH that is a big range of pH. So it could perform well also in slight acid soil, but also in alkaline soil. The pluviometric regime is about 700 millimeters per year. That is a common parameter in our region, and the annual mean air temperature from 8 to 15 degrees centigrade. For the chestnut, we know that is a species that is traditional here in the Apennines, so is well adapted to the climate condition and the soil condition, and it needs the sandstone parent material with sandy loam soil texture and pH that is acid, substantially acid or slightly acid. The pluviometric regime also like the hazelnut, so 700 millimeters per year, and the mean air temperature is the same as the hazelnut. For the soft fruits, because we have different species of soft fruits, so we have blackberries, currant, baby kiwi, and blueberry. We have different needed of parent material. So we have also a soil texture that is particular. So we will do some adjustment of the in the plantation for this species. Operatively, the research project is focused on the monitoring of the soil and their parameters with probes in the sites, and then we will make a soil sampling campaign at different times. So before the change of management or every kind of intervention, in the transition period, and then after the reaching of the full productivity period. And we will make physiochemical analysis, so laboratory analysis for have quantitative results. We will use also drones for providing information on the trees performance, on the plant performance, and we will use the VSA methods that I have explained you before for make expeditious screening between the main soil sampling campaign to check the results of the condition, the results of the intervention on the plantation. And then we want to use the GIS software to spatialize the available data. This is the references. And I thank you for your attention.
Thank you very much.
Thank you.
So in this case, it was a very nice presentation introducing all the main aspects of the importance of soil and the importance of cultivation system and soil management. Are there some questions?
Well, I have a question that is more, how can I say, a curiosity, but is related to the fact that there is an increased demand for growing hazelnut for the consumer demand and also for the role of Ferrero that is getting more and more space for hazelnut. And I wanted to know what is your point of view on this aspect in relation to the soil, especially those that are present in Marche region, and the potential for degradation eventually or preservation of the soil capacity.
My point of view is a very, very humble point of view. you know, also Beppe had said before in other circumstances that our region is not the best region to do hazelnut plantation. And in my opinion, if it will be, we have hazelnut plantation for from many years, you know. By the way, in our case study, we selected a soil that could perform, we hope, a good plantation with hazelnut, because the parameters are good for this kind of management. But this is not the rule for all the soil of our region. So I hope that the producer will ask to the researcher for help in this case.
In some way, you can also eventually take in consideration the possibility to have some correction or amendments to the soil, so to eventually solve the problem. Because in any case, I still see really difficult to grow hazelnut in our region, in particular in some area. Hopefully, in the areas that you selected, probably it will be more interesting, but more easy. But I think that it's really important to perform this kind of study and see what can be the real adaptability of hazelnut in that area. Because everybody, as Roberto say, everybody now is looking to hazelnut as a solution, as an alternative, but not really. Everybody knows that the soil is the main limiting factor in hazelnut cultivation.
Yeah, Bruno, to be part of the discussion. Sorry, Lorenzo.
No, it's not a problem.
Just to be part of the discussion. Yeah, this is true. We can, let's say, correct the soil to be cultivated with hazelnut. But the problem is that, you know, much more things you do on soil to prepare it to different cultivations then you will need also irrigation and preservation from pests. I mean, this as just a result to increase costs of production. So Roberto said a right thing, meaning what is the role of Ferrero? Ferrero now is really stressing the production of hazelnut. But they forgot till now, then we see in the future, they forgot to say that they don't put any money. They do not give 1 cent to the farmers to make any cultivation. So they only promise that in case you produce, they will buy the product. So this means that now they are promising from 3 to 6 tons per hectare of nuts. But we have some, models that say that probably in market region, we should be around 1.5. So this means that in front of an investment very high, because they also forgot to say that the proper cost for 1 hectare, considering fencings and irrigation, is from 16 to 20,000 euro per hectare. What they say, it is around 5 to 6, but they do not cite fencing and irrigation. So in front of a cost of an investment of 20 around euros per hectare, then you will probably produce something like 1.5 tons per hectare after 6 or probably 7 years. So by sure we can correct, and we can also change a little bit these numbers that I say now. But you know, from here to go toward convenience of investment, there is so much road to run, so much road. This is my, I mean, what according to the surveys that we had done and looking at what happened in the surrounding of market region or in similar source in other regions, for example in Umbria and others, boy, I would not suggest so much hazelnut cultivation in our region, but you know, everything is possible, I'm sure.
No, I fully agree. In fact, is what I was trying to say, that I think that we should give this message, a clear message, because growers get excited sometimes, but in reality, sometimes there is people not telling the truth, and so and that is the truth. I fully agree. So
thank you
it's
for
important that your study will help to understand better what would be the possibility. And so say, look, that is the real possibility. But then for all the others, say, look, be careful, because the situation is not in favor for that solution.
But I'm asking a naive question maybe, but would not be useful if, as a faculty located in Marche, we give advice, technical advice, nothing more, in order to make the farmer's choice easier, or is not needed because somebody else is doing it, I don't know, Assam or something like that?
You know, very difficult, Roberto, because the region, I mean, Marche Regional Administration has sort of, you know, they try to suggest hazelnut cultivation, also because pushed on it by the Ferrero. Also, Ferrero is doing really a very rotten.
We are not regional institutions. That's what I'm saying.
But it's also, well, we are going out from the discussion, but well, as far as I know, when people ask me something, I say, don't look at hazelnut, don't look at pomegranate, not look at all these exotic things that are exotic. The problem here is that around here, there are a group of people going at home with the grower and proposing to sell home at home directly this solution. And it's difficult to contract this people, because that they are dealers, are people that are paid or generated or created a small company with that pay. For sure, we have the role to say, to do this kind of study, publish, disseminate, and make aware of what is the real situation, so people can better decide on taking this culture crop in consideration or not.
Yes, and this is what we had done. Do, but sure. We had done. Sorry, just two words. We had done because in several occasions, I and others, we went around the region, say, "Boy, don't do it. Don't do it." Last time was in November at, I don't remember where, Steffi helped me. I don't remember where. I was attacked by a man saying, "Well, you are crazy, you cannot do this," because I was paid by Ferrero to stress hazelnut cultivation in the region, and you cannot destroy me. Okay, I say, thank you, guy, but so this is the situation, right, over the hazelnut cultivation. This is not easy, huh?
I agree.
I don't remember, this guy is one of, probably you know, name, I don't remember, Bruno, probably it's a big enterprise for machinery.
Yeah, I don't think, but I heard about. I think I heard about.
Yeah. So, yeah, and they had been paid, I guess, some 100,000 to stress this cultivation. So this is a very strange situation right now. Okay, sorry, just to be part of the discussion. Sorry.
But by the way, it's important to do this type of study, and also for the other crops that you mentioned about the berry, particularly blueberry and also raspberry. They will be linked to what Valeria will try to do. Most of the work will be done in soilless condition, because we know that also for raspberry, even if it's better than blueberry, the type of soils that we have here may have some difficulty. So it's a question for all this crop really to map all the different soils that we have, and say, okay, over there you can grow chestnut, you can grow hazelnut, you can grow blueberry and lenticchia and so on. So because all these crops are adapted to the acid soil. I fully support all this from my side. Okay. No other question? So I think that we can stop here today. Thanks also to Lorenzo that accepted to anticipate the seminar, so we can have the next section not so heavy. And please remember to your colleagues or other PhD students that are connected, please remember to send the missing titles, because we have some for some students still I don't have the title of the seminar. So send me the title of the seminar so I can complete the program. And we will be connected again next week, the 28th at 2 p.m. I say 2 p.m. because we have, in that case, 5 presentations, so it will be a more longer section. Actually, in that case, I still need 3 titles. Okay. Thanks very much.
Bruno, sorry, you said 28 next Friday, right?
Next Wednesday. Sorry, next Thursday, 28.
What is that? What about my calendar?
It's
Oh,
not-
it's Thursday. Yes. Okay. Good. Next Thursday, 2 p.m. Okay. Sorry.
Yeah. Then June 4, that is again Thursday. And June 11, that is again Thursday. So for the next 3 Thursday, if you are free, you are welcome to join us in this presentation, in this section. Okay.
Thank you, and bye-bye then.
Okay, bye-bye. Have a nice time to all.
Bye, thank you.
Thank you.
I thank you.
Ciao.
Bye.
Bye to you all.
Bye.
Bye bye.